Hello friends, welcome to Dr. Ossai Physiology Academy, DOPA for short. This is the place where we make learning of physiology easy, exciting, and effective. Thank you for joining me. And if you're new to this channel, you're especially welcome. And if you like the content that we share, kindly click the like button, click the subscribe button, and turn on notifications so you don't get to miss any new content we drop. All right, let's grow the channel together. So let's get right into today's lecture. Gastrointestinal secretion, one of the cardinal functions of the GIT. So now this is part 2. We're following it systematically. Okay, start from the mouth, stomach, intestines, small intestine, large intestine, and so on. So now we're done with the salivary secretion. So this is the part 2, gastric secretion, meaning the secretion from the stomach. So like we have said, the secretions are usually from glands. Okay, they are one of the factors that help in physiological processes, physiological functions. They implement what comes from the control center to modify, to control physiological processes. So in this case, now, first of all, we need to know the glands. You know, when you follow it systematically, it's easy for you. You need to know the gland that is secreting what. Okay? After knowing the gland, some peculiarities about that gland, especially the cells. What are the types of cells that are secreting particular substances? Then what are those substances? What are the composition? Okay, what's the composition of the substance? Then from the composition, you can now know the functions of the secretion in general. Okay, so it's easy. Then you now talk about the regulation. So we're going to be following it like that. So gastric secretion. The stomach, you know, it has, let's say this is the stomach that goes to that becomes the small intestine, the duodenum. So the stomach has the upper part, fundus. Then you now have the pyloric part around this side. Pyloric is the pyloric part. Okay, so you have the fundic glands, the pyloric glands. Okay, so now the stomach, this fundic part from the fundus, you have number 1, the oxyntic glands. Oxyntic glands. Then number 2, from this pyloric part, you have the pyloric glands. Okay, so we'll take it step by step. So within these glands, there are special cells that secrete the gastric secretions. So now let's start with the oxyntic glands. The oxyntic glands. Number 1, that's what we call mucous neck cells. But before I go that, something that will make it easier for us. You use this acronym, PHIM. There are 4 major secretions of gastric juice. You know, the gastric secretion, you also call it gastric juice. The pancreatic secretion also, you call it pancreatic juice. Those are the 2 that people like to call juice. All right. So this P represents pepsinogen. Pepsinogen. The H represents HCl. That's hydrochloric acid. The I represents intrinsic factor. Then this M represents mucus. Okay? So now, pepsinogen. It's secreted by peptic cells. Very easy to understand. So one of them, peptic cells. Okay, this is the gland, and these are the types of cells located in the gland. Secrete. You can also call these peptic cells chief cells. I don't know why they call them chief, but it's called chief cells. Then H, hydrochloric acid. Hydrochloric acid is secreted by parietal cells. Parietal cells. It's also called Oxyntic cells. Oxyntic cells. Then number 3, intrinsic factor, also secreted by the parietal cells. Okay? Intrinsic factor is also secreted by. Then the M, mucus, secreted by mucous neck cells. Mucous neck cells. All right? So this is Oxyntic gland from the fundic part of the stomach. So the pyloric gland from this side, it's not so difficult. Okay? This pyloric gland has some cells called G cells. G cells. They secrete gastrin. But gastrin is not a gastric secretion. It's not an exocrine gland. You know, when we're talking about all these gastric secretions, GI secretions generally, they are secretions that are secreted into ducts and they enter the lumen. Okay? But gastrin is a hormone in the sense that it is not secreted into ducts to enter the lumen to do anything. It is secreted into the blood. Hormones are secreted into the blood. So it is not part of the digestive function. A lot of all these cells, most of what they do is digestion and some other functions. Okay? Because like we said, all GI secretions, they contain mostly water, electrolytes, and enzymes. Okay? That's 3 basic things. What mainly differentiates them is these enzymes. The electrolytes are usually very similar, so we don't bother ourselves mostly with that. Then you also, this pyloric gland also has mucous neck cells. It has mucus, just like this one. Mucous neck cells that secretes mucus and bicarbonate. Okay? So you're wondering, parietal cells secrete hydrochloric acid. This one secretes bicarbonate, which is like alkaline. Why are they opposite? If you look at, you see that this one is at the fundus, the upper part. But this pyloric part, it has started secreting little bicarbonate. You will find out that the intestines mainly secrete a lot of bicarbonate. Of course, pancreatic enzymes, we'll talk about that later, but just to make you understand, pancreatic enzymes function in alkaline medium, while gastric enzymes function in acidic medium. So it's already going towards, so it's already preparing itself to start secreting bicarbonate to neutralize the acid. Okay? Because it's towards the pyloric side entering the duodenum. So don't get confused. So these are basically what you need to know. Use this acronym FIM to remember them. Okay? Peptic cells, pepsinogen, parietal cells, it secretes hydrochloric acid, also known as Oxyntic cells, an intrinsic factor, then mucous neck cells. So we are going to talk about the functions. Then very important is the mechanism of acid secretion, because one of the very important secretion has a lot of of impact in the stomach because it can cause ulcer, peptic ulcer disease, if you've heard of it before. It's related a lot to acid secretion. So we need to know the mechanism of acid secretion and all of that. So the next thing I'm going to talk about is the different functions of these secretions after this break. Welcome back. So going from the different composition of gastric secretion, let's now take the function. We can broadly divide the functions of the gastric secretion into two. One is digestive function. Digestive function. Number two, protective function. So digestive function is mainly from pepsinogen. And this pepsinogen is a protein hormone that digests protein. And protein, why protein digesting enzymes always come in the inactive form is the fact that a lot of the body's substance is made up of protein. No, we said that protein are the structural molecules. That's the introduction to the underlying principles to physiology. We mentioned that it's in my book. Okay, so that proteins are the structural and functional molecules of the cell of life. So the body is made up of protein. That's why when you eat meat, you say you are eating protein. Okay, animal body is made up of protein. So if this protein is in the active form, it can digest things that it should not digest. It can start digesting the body itself. So it needs to be secreted in the inactive form so that it has already come out into the place where it needs to function and before it's active. And there are protective mechanisms so that it will not digest what it should not digest. So digestive function is mainly by pepsinogen, which when it is activated, becomes pepsin. So what is the function of pepsin? Pepsin digests protein. So protein digestion starts from the stomach. Very important point to note. Protein digestion, but this pepsin is not as powerful as pancreatic enzymes and the organ. They do a lot of, the majority of digestion, but pepsin does some protein. So it digests proteins and converts them into peptones. Peptones and peptides. And proteoses. So these are intermediate molecules of protein digestion. It doesn't complete it. You know, when it's complete, it's amino acids. So it digests into peptone, proteoses, peptides. Okay? So that's the digestive function, proteins. Now, protective function of gastric secretion. It is carried out mainly by this mucus. Okay? The mucus, we introduced the term mucus in part 1, and we said that it's made up of mucin, very thick, slimy substance. So it helps to coat the surface of the stomach so that it protects it from the harsh corrosive action of hydrochloric acid. Okay? So it's mainly by mucus. The mucus secreted there protects the gastric mucosa from erosion from the hydrochloric acid. Then even the hydrochloric acid itself has protective function. How? Why? Now, remember that the food you are taking in from the outside environment, it is contaminated, no matter how you want to look at it. Sometimes you pick food to eat, your hand is not clean. You throw it inside your mouth. And even if your hand is clean, the food itself might be exposed to air for a long time. It's already cold and so on. The plate, the food you take is not at all sterile. It is loaded with microorganisms. So we said that the GIT is an extension of the external environment. But the nutrients will need to go into the internal environment. So one of the things that hydrochloric acid does is to kill microorganisms that can harm you. So it has bacteriolytic effect. Okay? So HCl has bacteriolytic effect. It kills bacteria and makes the food safe. Kills all those bad, offending microorganisms. Okay? Then, now let's talk about intrinsic factor. It doesn't have any function per se as it really relates to the GIT, but it has another function. It's secreted with hydrochloric acid, remember, from the parietal cells. So let's just put it aside. Intrinsic factor. Intrinsic factor. It helps in the absorption of vitamin B12. Vitamin B12, also known as cobalamin. Cobalamin. So cobalamin in itself, it's a maturation factor in the production of red blood cells. When red blood cells are produced, it's one of the factors that help red blood cells to mature in their developmental stages. So without intrinsic factor, if there's a problem with acid secretion, because they come together, parietal cells and intrinsic factor, there will be a problem with red cell secretion. It will lead to a form of anemia known as pernicious anemia. So lack of this leads to pernicious anemia. Okay? So that is basically what you need to know about the functions of gastric secretion. So now we are not going to go into the mechanism of acid secretion. It's very important that we know that. All right, so let's clean. So, mechanism of HCl secretion. Now, let's draw a parietal cell. Let's draw a parietal cell like this. Okay? So in a cell like this, you have the part that faces the blood. We call it the basolateral membrane. Okay? Remember the basic principle of secretion in the part 1 of the lecture. So this is the blood vessel. Okay? Capillary. Then this is the basement membrane. This is the capillary. This basement membrane. So this part is now known as the basolateral membrane. Then this part is the part that opens, that faces the lumen. So here is lumen. Here, this side is the blood side. Call it the blood side. Faces the blood. So this place is the apical membrane of the cell. Okay? This is the cell membrane, apical membrane. It's also known as luminal membrane. So what happens is that there's this very important common reaction that happens in a lot of cells. It's a reaction that has to do with water. Water is in the cell, the cytosol. And cells, through their metabolism, they produce what? CO2. Okay? So water combines with CO2 to give you something. And what is that thing? It's carbonic acid, H2CO3. Okay? That's what they combine to give. And this is catalyzed by carbonic anhydrase. The enzyme that catalyzes this. So what happens is that this carbonic acid is very unstable. It usually dissociates. It can go either ways, okay? It can dissociate and it gives you H plus and HCO3 minus. Now, this bicarbonate, HCO3, it will diffuse and enter into this blood. This is venous blood, to enter the venous blood. And it doesn't go alone. It will go in exchange for chloride ion. So there is a carrier that will do that. That will take chloride inside and take this bicarbonate, HCO3, outside into the venous blood. So this H that is produced, what happens is that it will now be secreted into the lumen. So H is secreted into the lumen with an enzyme, with a carrier that uses active transport. It is called hydrogen potassium ATPase. So look at it here. It will take potassium inside, it will take hydrogen outside. So hydrogen now is now in the lumen. So this chloride that comes from the blood, the chloride will come from the blood, enter into the cell, bicarbonate enters into the blood, exchange. Then the chloride that is now in the blood will now enter through chloride channels. Okay? Chloride channels. And you now have chloride. You have a lot of chloride, you have a lot of hydrogen, which they will mix together to give you HCl. That is how it happens. And there's something very interesting that also happens, is that this bicarbonate is alkaline, okay? So a lot of it enters into the venous blood, okay, of the stomach. That makes that blood very alkaline. It is called alkaline tide. Okay? It's called alkaline tide. It is noticed just after meals when you finish eating, because there's secretion of acid, a lot of acid. So it's like two opposing forces, a lot of acid into the lumen, a lot of alkaline into the blood. Okay? During meals, you get a lot of acid in the stomach, a lot of alkaline in the venous blood. But it's not going to waste. It's not just going to make this venous blood very alkaline for nothing. When we talk about pancreatic secretion later, you'll now see that pancreatic secretion needs a lot of this bicarbonate, because pancreatic enzymes, they need an alkaline medium. So it will go to the pancreatic blood supply and it is later. Okay? So this is the mechanism. It uses hydrogen, potassium, ATPase. Sometimes you call this proton pump. Proton pump. Because H is called proton. And because it's active transport using energy, ATP, it's called a pump. So this is the mechanism. Alright? Then lastly, we'll now talk about the regulation. Regulation of gastric secretion. Regulation of gastric secretion. Okay, so unlike the salivary secretion that only uses a nervous mechanism for its regulation, this gastric secretion uses both nervous mechanism and hormonal mechanism. Okay, so One, there are three substances that usually influence the gastric secretion, especially gastric acid. But when you're talking about gastric secretion, the main thing there is gastric acid. Pepsinogen is there too for protein digestion, but it's usually the acid that's more pronounced. Okay, so you have for the nervous mechanism acetylcholine. Acetylcholine. It is through a neurocrine. Neurocrine, call it a neurocrine mechanism through the nerves. Okay, so it happens a lot from the parasympathetic, is the parasympathetic branch of the autonomic nervous system that does that, influences gastric acid secretion. And sympathetic does what? Inhibits. Okay, just to digress a little, in the salivary secretion, both the sympathetic and the parasympathetic, they promote salivary secretion. But parasympathetic predominates, does more of it. But in gastric secretion, parasympathetic promotes parasympathetic. Why sympathetic inhibits minus this one? Plus promotes the secretion through a neurocrine mechanism, acetylcholine. Okay? So it promotes, it stimulates the parietal cells to produce acid, that reaction to occur. Then you now have what you know, the paracrine mechanism, which secretes histamine. So the histamine promotes, it stimulates the parietal cells also. Okay? So this paracrine, this histamine comes from a group of cells known as enterochromaffin-like cells. Let me write it here. Paracrine mechanism. Enterochromaffin-like cells. They're the ones that, so they use the H2 receptor. H2 receptor. That's the histamine receptor. Okay? It helps promote acid secretion. You know, H1 receptor is the one that has to do with the histamine there has to do with allergic reaction. So don't mistake it. This histamine, H2, its function is to promote acid secretion. So, number 3, what influences, stimulates the regulation? You have gastrin, secreted by the G cells in the stomach, but it's not directly into the parietal cells. It secretes into the blood, then it circulates and comes to influence the parietal cells. Okay? So gastrin secreted by the G cells to promote acid secretion. So these 3 things, acetylcholine, histamine from this one from G cells. Okay, these ones are from neurons. Okay, acetylcholine, parasympathetic, the vagus nerve, this neuron there. No, we said in the introductory lecture that the vagus nerve supplies 75% of the GIT. Okay, right from the esophagus down to some part of the transverse colon, only vagus. So this neuron is vagus nerve. If you want to reduce acid secretion, you can cut off the vagus nerve. It's called vagotomy. It's a treatment option. So that's just by the way. Right? So this is how it is controlled. And all these factors that control the secretion, what starts them, what stimulates them, and what can inhibit them, I will tell you. Now there are 3 phases of this stimulation to help in the regulation of this thing. Now, you have 3 of them, 3 phases. You have the cephalic phase. Very important for you to know. 1, cephalic. Cephalic phase. What do we mean by all this? You have the gastric phase of stimulation. Then you have the intestinal phase. Okay? So the cephalic phase is trying to tell you that from the sight of food, the smell of food, and even the taste of food, start stimulating gastric secretion even before the food gets there. That's what it means. And this level of stimulation it contributes is 20%. The cephalic phase. So the gastric phase is talking about, okay, when the food now arrives. Okay, when the food now arrives, the stomach. So this one, because we're talking of gastric secretion, it contributes the most, 70%. And this intestinal phase, talking about when the food is in the intestine. Usually it's inhibitory. This contribution is inhibitory because when it starts entering the acid chyme, enters the intestine, it starts inhibiting gastric secretion. So this one contributes 10% to influence gastric secretion, which is inhibitory. Okay? 20, 70, 10. So this is basically all you need to know at this level about gastric secretion. Okay? So for further reading, to get a better grasp of it in more details, I've written a book on gastrointestinal physiology broken down for you. This is it. And other textbook, 7 of them in physiology, as at the time of this shooting, okay? So just go to the description box. You see links there. You can download the soft copy from Okada Books just for a token for further reading. Also check, you see a link to our website so you can get to know some other things we do to make the learning of physiology easy, exciting, and effective. See you in the next video.